So this is chapter 27, where we'll be talking about fluid, electrolyte, and acid-base balance. We'll start with an introduction and talk about the three major types of balances we're going to discuss in this chapter. So we are essentially bodies of water. So most of our body weight is water. Water makes up 99% of our extracellular fluid, and it's also an essential component of our intracellular fluid. And we covered this earlier, but as a reminder, and we're going to go into more detail in this chapter, intracellular fluid is the fluid inside the cells. Extracellular fluid is the fluid outside the cells, and it includes like both the plasma in the blood and the interstitial fluid that is in the tissues between the cells. So our cells depend on water for the diffusion and distribution of gases, nutrients, and waste. So if we lose too much water, proteins can actually denature, which means they change shape. Enzymes stop functioning and cells will die. So the focus of this chapter is on the homeostatic mechanisms that regulate the volume, the ion concentrations, and the pH in these critical body fluids. So this chapter is going to deal with three main balances. The first is fluid balance, and you are said to be in fluid balance if the amount of water you gain each day is equal to the amount lost. Our digestive system is our main source of water gain, and the urinary system is the main route of water loss under normal conditions. You can also lose a lot of water when sweating, when your body temperature is elevated. Electrolyte balance is when the amount of each ion gained each day is equal to the amount lost. And electrolytes are ions released when inorganic compounds dissociate. They're called electrolytes because they have the ability to conduct an electrical charge in a solution. And we've been talking about these since the beginning of A&P. So electrolytes are things like potassium ions, sodium ions, and chloride ions, and other ions that have an electrical charge. The digestive system is our main source of electrolyte gain. Our urinary system is the main source of loss of electrolytes. Some electrolytes can also be lost in sweat and feces, but this amount is very small. Acid-base balance is when the production of hydrogen ions is precisely offset by their loss. So preventing a decline in pH, which is our body fluids becoming more acidic, is the body's primary problem because, as you're going to see later, we produce more acids than bases under normal natural circumstances. The kidneys and lungs play a major role in maintaining our acid-base balance. So, for example, if pH drops, it becomes more acidic, hydrogen ions can be eliminated by the kidney and carbon dioxide can be eliminated by the lungs. And we'll get into more detail with that later on. In this section, we'll talk about the fluid compartments, which is again going to be your extracellular fluid, which is mainly your interstitial fluid and your plasma, and your intracellular fluid, which is the fluid inside of the cells. So extracellular fluid, or ECF, is all of the fluid in the body found outside of a cell. There are two major divisions of the ECF. The plasma that makes up the liquid portion of your blood, and the interstitial fluid, which is the fluid in your tissues between all of your cells. However, the ECF also includes smaller amounts of other body fluids, so it would also include your lymph, your cerebrospinal fluid, synovial fluid in your joints, serous fluid in your body cavity, so this would be your pleural, pericardial, and peritoneal fluids, aqueous humor in your eye, and then perilymph and endolymph in the structures of your inner ear. Intracellular fluid, or ICF, is all of the fluid in the body found inside of the trillions of cells that make up our bodies. And so the fluid here, we talked about this back in chapter 3 when we talked about cells, but the cytosol is the fluid component of a cell's cytoplasm, and that is what is making up the ICF. So the ECF and ICF are considered to be fluid compartments because they behave as distinct entities. So if you look at the bottom of the picture up here, you have the intracellular fluid in yellow, and then you have your extracellular fluid over here on the left side made up of the plasma and the interstitial fluid. And so these two large compartments are separated by the presence of a cell membrane. So the cell membrane that is around each of our cells. So the fact that we have a cell membrane and the fact that we have active transport, so we can pump solutes in one direction or another, this allows the composition of the ECF to be very different from the ICF. So they can have very different electrolyte compositions. And we've been talking about this since the beginning of A&P. So if you remember, there's quite a few times all the way back since chapter 3 and many times since then where we've talked about how sodium concentrations are normally higher outside of the cell than inside the cell. And so that is a good example of an electrolyte difference between the ECF and the ICF. Water can move through most membranes by osmosis, so water will keep the osmotic concentration of each compartment identical most of the time. So generally they have the same osmolarity, but they can differ widely in their electrolyte compositions. So remember, water moves by osmosis based on total solute concentration. The concentration of individual solutes doesn't matter. And if you need a refresher on osmosis, this was covered back in chapter 3. So our bodies are mostly made out of water. So water accounts for 50 to 60% of our total body weight. And if you look down here at the bottom, adult males are shown on the left, adult females are shown on the right. So males have more water than females. Males are about 60% water. Females are about 50% water. And this is because males typically have a greater muscle mass and females typically have more adipose tissue. And if you didn't know, muscles are 75% water, while adipose tissue is only 10% water. And so males tend to have more muscle mass and less adipose tissue, therefore they have a greater percentage of water. However, it doesn't matter if it's a male or a female. In both sexes, the intracellular fluid contains a lot more water than the extracellular fluid. So if you look at this graph up here at the top right, there is more water in the intracellular fluid than there is in the extracellular fluid. And then if you look at the solid components that make up our bodies, proteins dominate. And again, that shouldn't be too surprising since proteins do all of the jobs in the cells. They are the workhorses of our cells. They make up the enzymes, structural proteins, and etc. So they make up the largest solid component of our bodies. So, as I mentioned, the ECF and the ICF can have very different electrolyte or solute compositions. So in this graph down here, we're showing the ICF on the left. We're also showing the plasma in the interstitial fluid, but keep in mind that both of these are part of the ECF. So the principal ions in the extracellular fluid, and again, it doesn't matter if it's plasma or interstitial fluid, it's the same, are going to be sodium, chloride, and bicarbonate. And again, this should be a bit of a review for you because we have talked several times about how sodium levels are highest outside the cells. Well, ECF is outside of the cells. The principal ions in the intracellular fluid or inside of the cells are going to be potassium. Again, that shouldn't be a surprise. We've covered that significantly many times since the beginning of A&P. But two new ones for you, magnesium and phosphate ions are also high inside of the cells. The ICF also has a lot of negatively charged proteins, which are called anions. So a cation is an ion with a positive charge, and anion is an ion with a negative charge. And so all of these negatively charged proteins inside of our cells help keep the inside of the cell more negative than the outside. Again, that shouldn't be a surprise to you because we have covered that before. So much of this should be a review. We started talking about the differences between the inside and the outside of cells from the very first unit of A&P 1 back in chapter 3. We've talked about it a lot since then. It was the basis for how we talked about how electrical potentials traveled through neurons. We talked about it earlier this semester when we talked about graded potentials. So again, a lot of this should be a review. And again, remember that water moves by osmosis based on total solute concentration. The concentration of individual solutes doesn't matter. So osmosis between compartments, between the ICF and the ECF, will keep the total osmolarity the same between the ICF and ECF, even though they have different subsets of solutes. So now we'll talk about solute exchange between the ECF and the ICF, as well as an overview of regulation of fluid and electrolyte balance, and then the primary hormones regulating fluid and electrolyte balance. So the ICF and the ECF can have very different concentrations of individual solutes like sodium and potassium because the plasma membrane of all of our cells are selectively permeable. So solutes can enter or leave cells only through specific membrane channels or protein carriers, and that allows the body to keep very different solute concentrations in the ICF versus the ECF. So exchange between the ECF and ICF can only occur across a plasma membrane of a cell, which should make sense because you're talking about the fluid outside the cell and inside the cell. So the only way you can get from one place to the other is going through the plasma membrane surrounding the cell. And we've talked about several examples of this throughout A&P, but just to remind you and to give you two examples. Example 1, a glucose transporter would be a protein in the cell's membrane, and it would be required to move glucose across the membrane. So if a particular cell had a glucose transporter, then it could transport glucose between the ICF and ECF. If it did not have that glucose transporter, then it could not move glucose between the ICF and ECF. Another example you should be familiar with, the sodium-potassium pump helps to keep sodium and potassium levels very different on either side of a cell membrane. So for every one ATP molecule, this pump moves 3 sodiums to the outside and 2 potassiums to the inside, and that keeps the potassium concentration high in the ICF and the sodium concentration high in the ECF. So since the ECF, the extracellular fluid, has different subdivisions, there is also solute exchange between these areas. And again, the two major subdivisions of the ECF are the plasma and the interstitial fluid. And solutes move between these two areas primarily across the endothelial lining of capillaries, which we discussed extensively in chapter 21. So we talked about capillary exchange, and that is where you move water and solutes between the plasma, which is in the blood, and the interstitial fluid, which is out here. So you are moving it between the two subdivisions of the extracellular fluid. So if you remember, filtration pushes stuff out at the arterial end, and osmotic pressure brings stuff back in at the venual end. So this is a little summary down here showing the two main compartments. So we have the ICF is one of our large compartments, and then you have the ECF, which is made up of the interstitial fluid and the blood plasma. And it reminds you in this image that materials moving into and out of the ICF have to cross the cell membrane, and substances moving between the plasma and interstitial fluid have to cross capillary walls. So variations between the subdivisions of the ECF are minor compared to the differences between the ECF and ICF, and you can see that if you go back and look at that bar chart from earlier. And so for this reason, we typically compare the ICF to the ECF when focusing on fluid and electrolyte balance, and we don't pay as much attention to the two major subdivisions of the ECF. So this is going to be an overview of the regulation of fluid and electrolyte balance, and we're talking about fluid and electrolyte balance together because fluid balance is actually tightly linked to electrolyte balance, because if you change the concentration of electrolytes, you will cause water to move by osmosis, which will then change the fluid balance. So fluid and electrolyte balance are very tightly connected. So there are 4 principles concerning the regulation of these two balances. The first one is that all of the homeostatic mechanisms that monitor and adjust composition of body fluids respond to changes in the extracellular fluid, not in the intracellular fluid. So the body is monitoring the ECF and makes changes based on changes to the ECF. This should make sense to you because receptors can only monitor the environment outside of the cells, and these receptors are mainly monitoring the blood plasma and the cerebrospinal fluid. It should also make sense because all of the ECF is more tightly linked together than the ICF. Remember, ICF is the volume inside of cells, and we have trillions of cells. So if the volume or electrolyte concentration of a single cell changed, that doesn't mean that the other trillion cells are necessarily in trouble. And so it wouldn't make sense to try to monitor the inside of every cell. It's easier to monitor the extracellular fluid, or the ECF. The second principle is that there are no receptors that directly monitor fluid or individual electrolyte balance. So there are no like water receptors or individual electrolyte receptors. Receptors instead monitor plasma volume. So these are like the baroreceptors that can sense when the blood vessels are stretching. And they can also monitor the overall osmotic concentration, not individual electrolytes. Like we don't have sodium sensors. We do have total osmotic sensors, and these are types of chemoreceptors. So plasma volume and osmolarity are used as indicators of the overall fluid and electrolyte balance within the body. The third principle is that cells cannot move water molecules by active transport. Water only moves passively through aquaporin channels, and water will always follow changes in osmotic gradients. So this is movement by osmosis from an area of low solute concentration to an area of high solute concentration. There are no water pumps that can move water against the gradient by using ATP. So water only ever moves passively, and it only ever moves by osmosis toward an area of high solute concentration. So there's a little phrase that we use to describe this. We can say water follows salt. This basically is saying water moves toward higher solute concentration, and salt would be an example. And we saw this in chapter 26 with the nephron loop. We used little ion pumps in the ascending limb of the nephron loop to pump out sodium and chloride ions, and then because we did that, the water followed by osmosis in the descending limb. And the final principle is that the body's water or electrolyte content will rise if your gains exceed your losses, but the content will fall if your losses exceed your gains. And that one should just make common sense. So now we're going to look at the 3 major hormones that regulate fluid and electrolyte balance. Again, this should be a review. This has been covered in multiple chapters by this point, and if you get out of A&P without knowing what these 3 hormones do, then you are not paying attention. So the first one is antidiuretic hormone, or ADH. This is a hormone that promotes water retention. It is released by the hypothalamus through the posterior pituitary, and it's released in response to osmoreceptors. And again, remember, these guys are monitoring the osmolarity of the ECF. So the rate of ADH secretion will vary with the degree of change in the osmotic concentration. So as the osmotic concentration of the ECF increases, more ADH will be released. There are two major effects from ADH. The first one is that it stimulates water conservation at the kidneys by reducing water lost in urine. And the second is that it stimulates the hypothalamic thirst center to make you thirsty and to promote intake of fluids. The second major hormone is aldosterone, which promotes sodium retention. This is released by the adrenal cortex in response to either rising potassium or falling sodium concentrations in the blood, or in response to the renin-angiotensin-aldosterone activation pathway. So remember that in that pathway, the end product, angiotensin 2, triggers the secretion of aldosterone. And also, as a reminder, renin, which kicks off the RAAS pathway, renin is released under three conditions: whenever you have a decrease in plasma volume or blood pressure at the juxtaglomerular complex, whenever a decrease in osmotic concentration is sensed by the distal convoluted tubule, or whenever there is decreased sodium or increased potassium in the renal circulation. So aldosterone causes the kidneys to conserve or reabsorb sodium ions and secrete potassium ions into the urine. And again, you can think water follows salt. So because the kidney is conserving sodium ions, keeping the sodium ions means also increasing water retention, because more water will be reabsorbed because it moves by osmosis and is following the sodium ions. And the natriuretic peptides, ANP and BNP, these promote fluid loss. They are released by cardiac muscle cells in response to abnormal stretching of the heart walls. So they are released when the blood pressure or the blood volume are high. And the effects of ANP and BNP are to reduce your thirst and to block the release of ADH and aldosterone so that you increase your water and sodium loss in the urine. So again, as a review, and here's an image, the major hormones in the fluid and electrolyte balance are going to be ADH, which helps you retain water, so less water is lost in the urine. Aldosterone, which helps you retain water and sodium, and so more potassium gets lost in the urine. And then the natriuretic peptides, which help you to lose water and sodium in the urine. So this one does the opposite of these two. So now we're going to talk a little bit just about fluid balance, and we're going to talk about fluid gains and losses and fluid shifts between the ECF and ICF, as well as dehydration and hyperhydration. So a fluid balance is when the amount of water gained each day is equal to the amount lost to the environment. So this is a table provided by your textbook showing the sources of water gains and the ways that water is lost. So water is mostly gained through the digestive system, either in our food or as a liquid, and water is mostly lost through the urinary system in our urine. And we're going to talk about some of these other things on this chart in a bit. So again, water gains, most gains are through eating and drinking. So in this picture, we are looking at the ICF over here inside the plasma membrane of a cell, and we have the ECF over here. And so again, when we talk about water gains and losses, they are usually going to happen at the ECF, and then changes in the ECF can then affect the ICF, as we'll see in a bit. So most of the gains are going to be water absorbed in the digestive tract, which averages about 2,200 milliliters a day. You can also gain some water called metabolic water. And so this metabolic generation of water comes mainly from the mitochondria in our cells. So if you remember from AMP1, mitochondria take in oxygen and they use that oxygen to make ATP. And in that process of aerobic respiration, the oxygen gets converted into water. So you're essentially changing the oxygen into water. So you do have about a 300 milliliter water gain that comes from the activity of your cells making ATP. So water losses, the largest loss is through the urine, so about 1,200 milliliters per day lost in the urine. A very small amount is normally lost in the feces, typically only about 150 milliliters per day. The rest is lost through perspiration, and there are two types of perspiration. There is insensible perspiration, which we can't control. This is the evaporation of water across the epithelia of our skin and our lungs. And so think of this as water vapor that is lost by evaporation, and that can average about 1,150 milliliters per day. And then sensible perspiration is evaporation due to active secretion by sweat glands. So when you think about sweating, that is sensible perspiration. So the amount that you sweat every day can vary greatly from one person to another, depending on your activity level and the temperature, etc. The maximum rates of being able to sweat is a loss of 4 liters per hour, which shows why it is very important to consume more water when you are heavily sweating. Running a fever can also increase your water loss by 200 mils or more per day. And when you're on a diet, you need more water because you're actually using more water to break down the fat tissue that you're trying to lose when you are dieting. So if you remember, the two largest extracellular fluid subdivisions are the plasma and the interstitial fluid. And water movement between these areas occurs at the capillaries. So something that we did cover in previous chapters is edema. Edema is swelling that happens in the tissues when you have an abnormal movement of fluid from the plasma to the interstitial fluid. So this would be an abnormal movement of fluid from one subdivision of the ECF to another. So if you remember from chapter 21, edema can occur when you have an excessive volume filtered out at the arterial end of the capillary compared to the volume reabsorbed at the venial end of the capillary, and all of that extra fluid that is being forced out of the capillary is too much for the lymphatic system to handle. So again, this little picture will help you remember. Normally you have this amount of fluid that exits at one end of the capillary and is balanced by fluid that comes back into the capillary, but in edema this balance is offset and you end up having more fluid go out of the bloodstream and into the tissues, which causes swelling. So anything that can change hydrostatic pressure or the colloid osmotic pressure can cause edema. And again, if you forget what these terms mean or how they relate, go back and look at chapter 21 when we talked about capillary exchange. So pulmonary edema results from increased blood pressure in the pulmonary capillaries. So this would be an example of an increased hydrostatic pressure at the arterial end of a capillary. Generalized edema is seen in cases of advanced starvation. This is when you have a decreased blood colloid osmotic pressure from a reduced production of plasma proteins because you're starving and you just don't have the resources to make those plasma proteins. And then you can have different types of localized edemas, which are local areas of swelling and can have multiple causes. So for example, you can have damage to capillary walls, which happens when you get a bruise and that bruised area swells slightly. You can have constriction of regional venous circulation to an area, and you can also have blockage of lymphatic drainage from an area, and this causes a specific type of edema called lymphedema, which we talked about in chapter 22. So a fluid shift is the rapid water movement between the ECF and ICF in response to a change in the osmotic gradient. Remember, water flow cannot be controlled actively. Water will always move toward an area with a higher solute concentration. So let's look at two examples. So if the osmotic concentration of the ECF, or extracellular fluid, increases, and so for example, this could happen if you have a loss of fluid without a loss of electrolytes, then the ECF would become hypertonic, right? So higher osmotic concentration means it is hypertonic. So let's look at the picture from your textbook. So this is an example showing what happens if the ECF becomes hypertonic. So normally we have an osmotic balance between the ICF and the ECF, but in this case we have a decrease in ECF volume. So we lost fluid, but we did not lose electrolytes. So we took some of the fluid away. That made this side more concentrated. So this side then becomes hypertonic compared to this side. So water will move out of the cells and into the ECF to restore the osmotic balance. So water movement would be from the ICF to the ECF. So I told my A&P1 class to remember water always moves toward a hypertonic area. That's one way to remember this. Water always wants to move toward areas of higher solute. Now, the opposite condition we can also talk about, but keep in mind your textbook only shows that first example. The opposite example is if the osmotic concentration of the ECF decreases. So, for example, you have a gain of water, like you drink a lot of water, and you don't have a corresponding gain of electrolytes. Then the ECF would become hypotonic, so more water to solute. Water will then move out of the ECF and into the cells. So it would be the opposite of what we're seeing over here. Water would go from the right to the left and would go into the cells to help balance that difference. So water movement would be from the ECF to the ICF, and this could cause the cells to swell. So because the ICF contains a larger percentage of the overall water in the body, it acts as a water reserve to help balance any losses and gains to the ECF. And so like we just saw with these two examples, the first example, if the ECF loses fluid, then water can move from the ICF to the ECF. And in the second example, if the ECF gains a lot of fluid, water can move from the ECF into the ICF. So thus the ICF acts as a water reserve. So dehydration or water depletion occurs when the water losses exceed water gains. So the loss of water will cause the ECF to become hypertonic. So you're taking water away, which concentrates the remaining solutes, making that fluid compartment hypertonic. So to compensate for this, water would move out of the ICF and into the ECF in order to keep the osmolarity balanced. So water would move from the ICF to the ECF, and thus the cells would end up shrinking some. The longer the fluid loss occurs, the more body fluids in both the ICF and the ECF become more concentrated, meaning you've got more solute compared to the volume of fluid. And you end up with a condition called hypernatremia, which is an abnormally high sodium ion concentration. So you don't even have to be eating sodium to have this occur. Basically what is happening is you're taking away the liquid in the body, which is meaning that the sodium that was already there is becoming more concentrated. And hypernatremia is an increase in sodium concentration, not actually actual sodium content. There could also be a significant drop in your blood volume and blood pressure because you don't have enough fluid in your body. So symptoms of dehydration start pretty mild. You can be tired, have dark urine, have less frequent urination, dry skin or lips. As the severity increases, your blood pressure can drop. You can end up being lightheaded, having headaches or dizziness and muscle cramps. And then if it's still not treated, you can end up having convulsions, hypovolemic shock, loss of consciousness, and eventually death. So things that can cause dangerous dehydration include excessive perspiration, inadequate water consumption, repeated vomiting, and repeated diarrhea. So basically, the loss of fluid without replacing it, and just not drinking enough in general. So the physiological response to dehydration is for the body to increase the secretion of ADH and renin and to trigger the sensation of thirst. Clinically, if someone comes in and they have severe dehydration, the clinical treatment would be to administer hypotonic fluids, which would counteract the hypertonicity of the patient's ECF. So now we'll look at water intake and overhydration. So normally when you drink water, so let's say you're just drinking a normal 8-ounce glass of water, it will temporarily cause the ECF to become hypotonic because you're adding more water, which will dilute the solute that is present. So it becomes more diluted, which would make it hypotonic. But normally fluid would then move from the ECF to the ICF, so it would go into your cells. The cells would begin to swell, but this is usually quickly counteracted by the kidney, which will release the excess water in urine. So overhydration is typically not a problem. However, hyperhydration or water excess occurs when water gains rapidly exceed the water loss. So you're taking in more water than your kidneys can keep up with and helping you to lose. And so this is when it can become dangerous. So possible causes of hyperhydration are drinking large volumes of fresh water in a short period of time. And there is a video on D2L about the case of Jennifer Strange. She was the one who was doing that contest to hold your wee for a wee, and she ended up dying. So not only was she drinking a large volume of fresh water in a short period of time, but they were holding their bladders, holding their urination, and not getting rid of the water because that was the contest. You can also, if you get an infusion of a hypotonic solution into the bloodstream when you don't need one, that can cause hyperhydration. If you have the inability to eliminate excess water in your urine, so in cases of chronic renal failure, heart failure, or cirrhosis, that can cause hyperhydration. And then if you have an endocrine disorder like an excessive ADH production, which makes you hold on to your fluid and not get rid of it, that can cause hyperhydration. So with hyperhydration, the reason it becomes dangerous is the sodium concentrations in the body become abnormally low. So the sodium becomes too diluted with all of the extra water. And this is called hyponatremia, an abnormally low sodium ion concentration. And the most dangerous part of the hyperhydration is when the neurons in the CNS, the central nervous system, start to swell. So remember, fluid is moving from the ECF to the ICF in this case, which is going to cause the cells to swell. But the brain doesn't have much room to swell. And so this can start off by causing symptoms of drunkenness, which is referred to as water intoxication. But then it can rapidly progress to hallucinations, convulsions, coma, and then death, as happened with the case of Jennifer Strange. So the treatment for someone who has been hyperhydrated is to administer diuretics to promote water loss and a concentrated salt solution, so like a hypertonic solution, to counteract the hypotonicity of their ECF. So if she had gone to the hospital and gotten treatment, she might have survived, but they didn't know about this danger when they were doing this competition and she was drinking all that water. So now we'll talk about electrolyte balance, and we're really only going to focus on sodium and potassium. So electrolyte balance is when your electrolyte gains and losses are equal, but it applies to each individual ion. So for example, you would be in sodium balance when your sodium gains and sodium losses are equal. So the importance of electrolyte balance, we've already kind of mentioned this before. The total electrolyte concentration can directly affect water balance because water moves by osmosis toward areas with high solute or electrolyte concentration. So the concentrations of individual electrolytes can also affect cell function, so it's important for each individual electrolyte to be in balance as well. So we're going to pay close attention to sodium and potassium because first, they are the major contributors to the overall osmolarity of both the ECF and the ICF, and they directly affect the normal functioning of all cells. Like, for example, when we talked about neurons, sodium and potassium are both essential ions for generating action potentials. They're also important for muscle contractions and the functions in several other cell types within the body. So in general, the most common problems with electrolyte balance are caused by sodium, but the less common but more dangerous problems are caused by potassium. So remember that sodium is the main cation or positively charged ion of the ECF. So again, to remind you, sodium concentration is high on the outside in the ECF, and potassium is the main cation of the ICF. So potassium concentrations are highest on the inside of the cells. So sodium gains typically happen through the digestive system. So we gain sodium through the food that we eat and the drinks that we drink. And sodium losses typically happen through the urinary system. So we lose sodium in our urine. Some sodium is also lost in perspiration, but only very small amounts. When you eat a meal high in sodium, so you throw in a lot of extra salt in your food, that extra sodium is going to get absorbed through the digestive tract, and it's going to go into your ECF. So when it goes into your bloodstream, remember that plasma is part of the ECF. And so this is going to cause more water to move into the ECF to balance out the osmotic concentration. This is why eating a lot of salt can cause high blood pressure, because the salt goes into the blood plasma, water will follow to equalize the osmotic concentration, and adding water to the plasma is going to cause a higher blood volume, which is going to lead to a high blood pressure. So this is why if you have high blood pressure, the doctor is probably telling you to try to cut salt out of your diet. Sodium regulation can happen indirectly by changes in blood volume and blood pressure from fluid shifts that are caused by the sodium gains or losses. And again, keep in mind, water follows salt. So where the sodium goes, water will follow behind. So let's look at two examples. These should still be reviews from stuff we've covered earlier. So if you have a gain in sodium, that is going to cause the ECF volume to increase because water follows salt. So more salt means more water. So you have an ECF volume gain, which leads to an increased blood pressure. This would cause a release of the natriuretic peptides from the heart, and these would promote the water and the sodium loss. On the flip side, if you're losing too much sodium, so you're losing more sodium like in your urine than you're eating, your ECF volume will become less. So again, water follows sodium, so less sodium means less water, means less volume. This would lead to a decreased blood pressure. This would lead to a release of renin and ADH, which would then promote sodium and water retention. So again, we covered this earlier. Your ADH and aldosterone is going to promote you keeping water and sodium in the body, and your natriuretic peptides are going to promote the loss of water and sodium in the urine. So sustained problems with sodium balance usually only occur when there is also a fluid imbalance that is happening at the same time. So like we talked about earlier, hyponatremia can happen when you have too much fluid, like in the cases of overhydration. And hypernatremia, or too much sodium, can happen when you have too little fluid, like in the cases of dehydration. 98% of all of the body's potassium is inside of our cells. So again, potassium is mainly in the ICF. We typically gain potassium through the digestive system, and we lose potassium through the urine. Remember, though, that electrolyte balance is based on concentrations in the ECF because that's where the receptors are. So the potassium concentration in the ECF, even though it only represents 2% of the body's total potassium, that is the concentration that is regulated, and it's regulated by making adjustments to how much potassium is lost in the urine. So the rate of tubular secretion of potassium into the urine depends on 3 factors. First, as the potassium concentration of the ECF increases, the secretion of potassium increases, so you would end up with more potassium in the urine. When aldosterone is present, the secretion of potassium also increases because remember, aldosterone causes you to reabsorb sodium, but in order to do so, a sodium ion is exchanged for a potassium ion, so therefore potassium would be lost in the urine. And when the pH of the ECF decreases, the secretion of potassium into the urine decreases, and this is because when you have a low pH, as we'll look at in a minute, the kidney will secrete hydrogen ions, but the hydrogen ions take the place of the potassium ions in those particular transporters. So when you have to get rid of hydrogen ions by the kidney, naturally that means you're going to hold on to more potassium in the body. So there are 2 main conditions that we have to worry about with potassium: hypokalemia and hyperkalemia. So hypokalemia is caused by too little potassium in the blood. This can be caused by too much aldosterone. So remember, aldosterone causes sodium retention and potassium loss. So too much aldosterone can cause you to lose too much potassium. And diuretics. So diuretics can increase the volume of urine you produce, and so that can inadvertently also cause you to end up with hypokalemia. Hyperkalemia is when you have too much potassium in the blood. This can be caused by acidosis, which we're going to talk about in the next section. And so with chronically low blood pH or acidosis, this promotes hyperkalemia by interfering with the potassium excretion. So again, in those cases, the kidney stops secreting potassium and secretes hydrogen ions instead. Kidney failure or damage to the kidneys can also prevent normal potassium secretion and lead to hyperkalemia. And if you take drugs that block sodium reabsorption, then blocking that sodium reabsorption slows down, so that will also slow down the potassium secretion because those 2 are linked as well. And the reason why potassium levels are so important is because if you have too much or too little potassium, both directions can affect the heart. So severe hypokalemia is lethal, and severe hyperkalemia can cause cardiac arrhythmias. So a lot of people think that when you exercise, you have to also take electrolytes in addition to drinking water. And you actually don't lose as many electrolytes in your sweat as people think. Sweat is actually a hypotonic solution containing more than 99% water and only very small amounts of sodium and other electrolytes. And someone who is sweating a lot is actually losing far more water than they are losing sodium or other electrolytes. And the loss of water actually increases the sodium concentration of the ECF. So even though you may be losing a little bit of sodium, because you're losing so much water, you're actually concentrating the sodium left in your body and increasing that sodium concentration. So the best thing to do when exercising is to drink plenty of plain water. There's no scientific evidence that drinking plain water while exercising will cause cramps, which is usually the reason cited by most people to not drink plain water during exercise. And your body has enough electrolytes in reserve to tolerate extended periods of strenuous activity, so you don't need to have electrolytes in the water you are drinking. So some of the problems with sports drinks, and sports drinks are drinks that contain sugars, vitamins, and electrolytes in addition to the water, is that the high sugar concentrations in these drinks can actually cause cramps, diarrhea, and other issues. And because they put vitamins in a lot of them, you do not lose vitamins when you're exercising. So if you're drinking a lot of these sports drinks, you can end up actually having an overdose of certain types of vitamins. So we focused on sodium and potassium, which are shown here. This is the chart from your textbook. So again, we talked about hypernatremia, hyponatremia, and then hyperkalemia and hypokalemia. Your textbook also goes into a little bit more detail with the other electrolytes like calcium and magnesium. We've actually talked quite a bit about blood calcium regulation in past chapters, so I am not going to revisit it here, but there is also information on the causes of hypo and hypo conditions in both calcium and magnesium, as well as phosphate and chloride ions. You can also see what signs and symptoms of these hypo or hyper conditions entail, what can cause them, and what the treatments are. So this is provided more for your information, but if I'm going to test you over anything, it would just be over the sodium and potassium information that we covered. So now we're going to look at acid-base balance, and we're going to start off by introducing pH and talking about the types of acids produced by the body. So pH is a measurement of the hydrogen ion concentration in a solution. So when we look at the pH scale, we are basically looking at a relative measurement of how many hydrogen ion concentrations are around inside of a solution. So the more hydrogen ions that are present, the more acidic a solution is, and the lower the pH. So as the pH number gets smaller, your solution is becoming more acidic, and that means you have a higher level of hydrogen ions. The fewer hydrogen ions there are, then the more alkaline a solution is, and the higher the pH. So as the pH number gets larger, your solution becomes more basic or alkaline, and that is because you have a lower level of hydrogen ions. And did you know that hydrogen ions can also just be called protons, because a hydrogen atom is made up of a single proton and a single electron, and that electron gets taken away to make the ion. So essentially a hydrogen ion is just a single proton. So an acid is a substance that adds hydrogen ions to a solution. So acids would make the pH become smaller. And bases are substances that remove hydrogen ions from a solution, so they would make the pH higher. So basic means the same thing as alkaline. So the acid-base balance in the body is the homeostatic mechanisms for controlling our physiological pH. This is very important for the proper functioning of an organism. Changes in the hydrogen ion concentration can disrupt the plasma membrane stability. It can change the structure of proteins, and it can inactivate or alter the functions of our enzymes. The pH of the extracellular fluid normally ranges between 7.35 and 7.45. So notice that this is a very narrow range. So there's not a lot of wiggle room for the pH in our body. This is also slightly alkaline, so 7 is considered neutral. So this is slightly on the alkaline side of neutral. If the pH falls below 7.35, that is called acidemia, and that results in the condition called acidosis. If the minimum pH that you can survive is 6.8, pH falls below that, death will occur. If pH rises above 7.45, that is called alkalemia, which results in the condition called alkalosis. and the maximum pH that we can withstand is 7.7. If you get higher than that, death occurs. So to give you some examples of how pH can really disrupt the functioning of the body, let's look at examples of what can happen if your pH falls below 7, which would be considered to be a severe acidosis. Your central nervous system function would deteriorate and result in a coma. Your cardiac contractions, so the contractions of your heart muscle, gets weak and irregular and can result in heart failure. And you end up having peripheral vasodilation, which would cause a dramatic drop in blood pressure and can result in circulatory collapse. So maintaining a proper pH is very important. So acidosis and alkalosis are equally dangerous, but problems with acidosis are more common because our body naturally produces several types of acids. So we're going to look at the 3 major types of acids produced by the human body. So we have 3 main categories of acids. The first is fixed acids. These are acids that do not leave a solution. So in other words, they stay in our body fluids until the kidneys secrete them. And these are acids that are also generated during the catabolism or breakdown of amino acids and other compounds that contain phosphate groups. The 2 largest examples of fixed acids are sulfuric acid and phosphoric acid. There are also metabolic acids, which are participants in or byproducts of cellular metabolism, so aerobic metabolism. Examples of these are pyruvic acid, also called pyruvate, lactic acid, and ketone bodies. Under normal conditions, these compounds are metabolized rapidly and don't accumulate in the body. And then you have volatile acids. They are volatile because they can leave the fluids in the body and enter the atmosphere via the lungs. And the major volatile acid we make is carbonic acid, which is formed from carbon dioxide. So remember, carbon dioxide and water can be joined together to make carbonic acid, which can then dissociate into hydrogen ions and bicarbonate ions. So actually, the amount of carbon dioxide in our body is the most important factor that affects the pH of our blood and the rest of the ECF. So remember that carbon dioxide can be ultimately converted to hydrogen ions, and then also remember, as you add hydrogen ions, you decrease the pH. So remember this equation that I said was never going to go away. Here it is again. So if you take water and combine it with carbon dioxide, you get carbonic acid. And remember, carbonic anhydrase is the enzyme that catalyzes this reaction. And then the carbonic acid can then dissociate into hydrogen ions and bicarbonate ions. So there is a direct link between carbon dioxide and hydrogen ions, which, if you remember, hydrogen ions are what we're measuring when we measure pH. So there's a direct link between carbon dioxide and pH. So carbon dioxide and pH are inversely related. So the more carbon dioxide you have, the more hydrogen ions you make. But remember, go back and look at that pH scale. The more hydrogen ions we have, the smaller the pH number. So more carbon dioxide, more hydrogen means a lower pH. Conversely, if we have too little bit of carbon dioxide, we actually use up some of our hydrogen ions to make more carbon dioxide. So a decrease in carbon dioxide means a decrease in hydrogen ions, which would be an increase in pH. So they are inversely related. And in addition to remembering the pH values of between 7.35 and 7.45, you should also remember, this was covered in chapter 23, the normal range of carbon dioxide partial pressures in our blood should be between 35 millimeters mercury and 45 millimeters mercury. This is acid-base balance part 2, where we're going to look at buffer systems and then respiratory and renal compensations for changes to pH. So to regulate the body's pH, the body has to balance its gains and losses of hydrogen ions. But the places where hydrogen ions are eliminated, which is mainly going to be the lungs and the kidney, are far removed from the sites of production. So they can be produced in cells throughout the body. And so the hydrogen ions have to be transported through the body to get them from their site of production to the site where they can be eliminated. And this requires the presence of buffers to neutralize these acids and to protect the tissues as they travel through the ECF to get to the site of elimination. So a buffer is a substance that can oppose pH changes by either removing or adding hydrogen ions as needed. So buffers help to keep the pH stable. So that way, if you have a lot of pH or you have a lot of hydrogen ions in your blood that you need to eliminate and you're sending it to the kidneys, in the meantime, you don't want the blood's pH to drop. And so the buffers help to prevent those pH changes until you can get rid of your excess hydrogen ions. So here is a graphical example. So a buffer has the capacity to both remove hydrogen ions or add hydrogen ions as needed to the solution around it. So if we have a condition where the solution becomes too acidic, so our solution has too many hydrogen ions, we need to get rid of some of those so they're not lowering the pH. So a buffer would have the ability to absorb or bind to hydrogen ions, which removes them from the solution and helps stabilizes the pH to normal. So in this picture, these are the hydrogen ions floating around in our solution. And so when they become attached to the buffer through a chemical bond, they are no longer free-floating hydrogen ions, so they can no longer have an effect on the pH. On the flip side, if the solution becomes too basic, meaning we don't have enough hydrogen ions in our solution, a buffer can release hydrogen ions and add them to a solution. So they donate or release hydrogen ions, adding them to the solution. That puts more free hydrogen ions in the solution, which will decrease the pH back to normal. So a buffer is called a buffer because it can do the reaction in both directions. It can bind to hydrogens or it can let them go as needed to change the concentration of hydrogen ions in the solution. So a buffer system is usually composed of a weak acid and its conjugate weak base. And it's usually written in an equation like this, where you have the hydrogen ion attached to the molecule here, and you have a reversible equation where it can become unattached, and it would just be a free-floating hydrogen ion. So the body has 3 major buffering systems, and we're going to look at these in more detail, but here's an overview. So the phosphate buffer system is going to regulate pH primarily in the ICF, or the intracellular fluid. The bicarbonate carbonic acid buffer system is going to regulate pH primarily in the ECF, or extracellular fluid. And then we have protein buffer systems, which can regulate pH in both the ICF and the ECF. So let's look at the phosphate buffer system first. The phosphate buffer system, as we just said, is important for buffering the intracellular fluid. It can also be used to stabilize the pH of the urine produced in the kidneys, but we're not going to go into a lot of detail with that in this chapter. And although you do have the phosphate buffer system present in the ECF, its role is superseded by the bicarbonate buffer system because you have much more bicarbonate ions than you have phosphate ions in your ECF. So with the phosphate buffer system, hydrogen ions can be passed back and forth from a compound called dihydrogen phosphate, and then this dihydrogen phosphate can release a hydrogen to form monohydrogen phosphate. And then so it can go both directions. So if you need hydrogen ions, you can create them. If you need to get rid of them, you go back this way and make dihydrogen phosphate. So cells can maintain a phosphate reserve by keeping around sodium monohydrogen phosphate, which can then be used to provide more phosphate ions for this buffer system. So if you combine sodium with monohydrogen phosphate, you get sodium monohydrogen phosphate, which can then be reversed to give you more monohydrogen phosphate, which is part of this buffer system. The next buffer system are the protein buffer systems. So remember, proteins are macromolecules made up of subunits called amino acids. And amino acids have the ability to accept or release hydrogen ions, which make them buffers. So because proteins are everywhere, inside and outside of cells, the protein buffer system will play a role wherever you have proteins present, meaning that it plays a role in both the ICF and the ECF. So let's take a closer look at amino acids acting as buffers. So amino acids really have three main parts. They have an amino group, they have a carboxylic acid group, which is their acid side. They also have an amino group, that's where the amino acid comes from, and then they have a variable side chain, which is represented by the letter R. So the carboxyl group, which is the acid part of the amino acid, can bind to additional hydrogen ions when needed. So if you start here in the middle where we have a neutral pH, the carboxyl group can accept a hydrogen out of solution where it can bind right here, and that takes the hydrogen out of the solution, and so that is going to help buffer extra hydrogen ions. And then the amino group can release its hydrogen ion when needed. So again, if you start here in the middle, so if we need more hydrogen ions because we have an increase in pH, this group can let go of that hydrogen ion, which can help to lower the pH back to normal. So basically, amino acids can help changes in either direction, which make them buffers. And then there are some side chains or R groups that can also donate or accept hydrogen ions, which further reinforces their buffering capability. Hemoglobin is a protein in our red blood cells, which also acts as a buffer system. So as we covered in chapter 23, hemoglobin can bind directly to hydrogen ions and thus buffering changes in pH. And hemoglobin can also secrete bicarbonate ions through the chloride shift, which adds bicarbonate ions to the ECF. So this is a portion of the picture from chapter 23. So just as a reminder, inside the red blood cells, after we form carbonic acid, it dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions are actually buffered by hemoglobin itself, and the bicarbonate ions can move out of the red blood cell and into the ECF in exchange for a chloride ion, and that was called the chloride shift. And then the carbonic acid carbonate buffer system is the third of the three buffer systems. So keep in mind that every cell in your body, almost every cell in your body, is generating carbon dioxide every second of every hour of every day, because carbon dioxide is generated by the mitochondria as a byproduct of aerobic respiration, where they are using oxygen to make ATP. So the majority of the CO2 produced by our body is converted to carbonic acid. So here's this familiar equation again. Carbon dioxide and water combine to make carbonic acid. This reaction is catalyzed by the enzyme called carbonic anhydrase, and then the carbonic acid then dissociates into hydrogen ions and bicarbonate ions. So a change in concentration of any component of this reaction can shift the direction of the reaction. So you can shift it in one way to take away hydrogen ions and shift it in the other way to add hydrogen ions, which is what makes it a buffer system. So for example, if you had an increase in hydrogen ions, that would shift the equation to the left towards the carbon dioxide. So you would be making more carbon dioxide, which could then be exhaled through the lungs. So remember, carbonic acid is an example of a volatile acid. So there are 3 limitations to this buffer system. This is the buffer system that primarily buffers the ECF. The first limitation is that it cannot protect the ECF from changes that result from altered carbon dioxide levels. So because the equation depends on carbon dioxide, if you have a problem that is affecting the carbon dioxide, you're going to mess with this buffer system. So since it depends on carbon dioxide, changes in carbon dioxide will disrupt this buffering system. Number 2, it can only function when the respiratory system is functioning normally. So it depends on the ability to exhale extra carbon dioxide in order to rid the body of extra acid. So if you have a respiratory system problem, this buffer system will not be as functional. And number 3, its ability to buffer acids is limited by the availability of bicarbonate ions. So when the equation does shift to the left and you use the taking away the hydrogen ions from the solution to make carbonic acid, that requires that you have a bicarbonate ion to attach to them. So it does depend on the availability of bicarbonate. Now, the body does contain large reserves of bicarbonate ions in the form of sodium bicarbonate. So this is called the bicarbonate reserve, and it can add more bicarbonate ions when needed. And then the kidney also has the ability to create new bicarbonate ions from carbon dioxide in the DCT and the collecting system. And as they do this, the hydrogen ions are immediately secreted into the urine. So it is just adding bicarbonate ions to the body. So this figure from your textbook reviews the carbonic acid carbonate buffer system. So again, we have the conversion of carbon dioxide and water to carbonic acid, which can dissociate into hydrogen and bicarbonate ions. So the bicarbonate ions can be put into reserve by combining them with sodium to create the sodium bicarbonate. And remember that these equations can move in both directions. So in the case of where you have the presence of acid, so you're adding hydrogen ions, you can pull up the bicarbonate ions from your reserve. You can use those bicarbonate ions to connect them with the hydrogen ions to make an increase in carbonic acid, which you can then convert to carbon dioxide and exhale through the lungs. So that is a way that the body can get rid of acid or hydrogen ions. So this system is primarily responsible for buffering pH changes that occur as a result of the production of fixed acids or metabolic acids. And again, the carbonic acid carbonate buffer system helps to buffer changes in the extracellular fluid compartment. So buffer systems help to momentarily tie up extra hydrogen ions, but the body still has to get rid of them. So remember, the buffer system is there to help prevent pH changes while you are transporting the hydrogen ions to the point where you can get them out of the body. So buffers only render hydrogen ions harmless until they can be excreted. So there are 2 primary ways that the body gets rid of extra hydrogen ions, and that is through the respiratory system and the urinary system. So in respiratory compensation, the respiratory rate can change to stabilize the pH of the ECF, and chemoreceptors in the ECF are sensitive to changes in carbon dioxide levels. So respiratory activity, therefore, can have a direct impact on the carbonic acid bicarbonate buffer system. So as the concentration of carbon dioxide increases, remember that means we're getting an increase in hydrogen ions, and that will cause a decrease in pH. So the compensation for this is to increase the respiratory rate, which helps to remove the carbon dioxide, which is going to help remove hydrogen ions and increase the pH back to normal. The opposite change can also happen and be compensated for. So as the carbon dioxide level decreases, you have a decrease in hydrogen ion concentration, which will cause an increase in pH. So the compensation for this would be to decrease the respiratory rate, so you slow down your exhalation of CO2, and this allows CO2 to start building up in the body, so you retain and increase your amount of CO2, which will allow you to have more hydrogen ions, which will decrease the pH back to normal. And then with the renal compensation, you can change the rates of hydrogen and bicarbonate, either the secretion of these ions or the reabsorption of these ions in the nephrons of the kidney. So in response to a decrease in pH, which is a condition of where you are more acidic and you have too many hydrogen ions, the kidneys can increase their secretion of hydrogen ions into the urine and increase their reabsorption of bicarbonate ions back into the blood. So the kidneys also have their own buffering systems for the tubular fluid, which allows them to secrete more hydrogen ions because they're able to buffer them while they are in the tubular fluid, which lets the kidney secrete more hydrogen ions than they would otherwise be able to. And in response to an increase in pH, which would be a less acidic condition, so you don't have enough hydrogen ions, the kidney can compensate by reabsorbing hydrogen ions and keeping them in the blood while secreting bicarbonate ions into the urine. So here is the response of the lungs and the kidney to acidosis. Acidosis would be a decrease in pH. So if we start up here at the top, if we have the condition of acidosis, that means we have too many hydrogen ions. So there are two ways to get rid of these hydrogen ions. One way is for the hydrogen ions to be converted into carbonic acid by joining them with a bicarbonate ion. And if you need a bicarbonate ion, you can get one from the bicarbonate reserve, and the kidneys can also contribute bicarbonate ions. You combine the hydrogen ion with the bicarbonate ion to make carbonic acid. Carbonic anhydrase then converts that into carbon dioxide and water. The carbon dioxide is thus exhaled, and so that gets rid of that hydrogen ion because it is now stuck in water. And then there are other buffer systems that help to buffer that hydrogen ion until it can get to the lungs or until it can get to the kidneys. So what the kidneys can do in this condition is they can secrete hydrogen ions into the urine, which helps to take them out of the body. And they can also reabsorb bicarbonate. And then the response to alkalosis, which would be an increase in pH. And in alkalosis, we don't have enough hydrogen ions. So basically, it's the opposite of what was above. So now we don't have enough hydrogen ions, so we can slow our breathing rate. If we slow our breathing rate, that allows CO2 to build up in the body. As that CO2 builds up, it can be combined with water to make carbonic acid, and then the carbonic acid can provide us with more hydrogen ions. Buffers can also release their hydrogen ions, and the kidney can secrete bicarbonate ions and conserve hydrogen ions to also add to the hydrogen ion supply, and that will help to lower the pH back to normal. And now we'll look at disorders of acid-base balance. So we'll talk about respiratory versus metabolic disorders and acidosis versus alkalosis. So conditions that can cause problems with acid-base balance include any disorder that affects the buffer systems, the respiratory performance, or renal function. So since all of these systems help to maintain a normal pH, dysfunction in any of these areas can cause issues. So for example, both emphysema, which would be a failure of the lungs, so a failure of respiratory performance, or renal failure, which would be a failure of kidney performance, can cause serious pH problems. Also, cardiovascular conditions can affect acid-base balance. So changes in the cardiovascular system can cause fluid shifts between the ICF and ECF. It can also change the glomerular filtration rate of the kidney, and it can alter respiratory efficiency. So, for example, both heart failure or hypotension, which is abnormally low blood pressure, could cause serious pH problems. And third, central nervous system conditions can also cause problems with acid-base balance. So the central nervous system controls both cardiovascular and respiratory reflexes, and these reflexes are essential to normal pH regulation. So damage to those areas of the brain can also cause serious acid-base balance problems. So most acid-base disturbances have two phases. The first phase, or the acute phase, is the period when the pH is moving rapidly out of the normal range. And the compensated phase is the period that occurs after physiological adjustments have kicked in. So unless the underlying problem is fixed, though, the compensatory mechanisms are usually unable to return the pH to normal ranges. They're kicked in and they're trying to fix the problem, but they can't take you back to normal unless you fix the underlying problem that is causing the pH imbalance in the first place. So now let's look at respiratory acid-base disorders. So respiratory acid-base disorders occur whenever there is an abnormal carbon dioxide level in the extracellular fluid due to breathing issues. So the first of these is called respiratory acidosis, and this is actually the most common acid-base disorder. So respiratory acidosis is characterized by low plasma pH and elevated plasma carbon dioxide levels because the respiratory system is unable to eliminate all the carbon dioxide that is being generated by peripheral tissues. Remember, every cell of the body is making carbon dioxide every second of every day. So hypercapnia is the term that refers to elevated levels of carbon dioxide in the blood. The usual cause of respiratory acidosis is hypoventilation, which is an abnormally low respiratory rate. The problem can become quite severe in a short amount of time. Even just a few minutes of stopped breathing can cause CO2 levels in the body to build up to dangerous levels because, again, every cell in your body is making CO2 every second of every day. So acute respiratory acidosis is an immediate life-threatening condition that is caused by a complete lack of being able to breathe for a while, like during a heart attack or a drowning victim or someone who is choking. The reversal of this acute phase is a major goal in resuscitation of cardiac arrest or drowning victims. And this is why the ABCs of emergency care, the first two letters is airway and breathing. So the first thing you focus on is getting them to be able to breathe again. Chronic respiratory acidosis occurs when you have normal respiratory function is compromised due to some condition. So this can be caused by anything that slows the normal rate of breathing. So some examples could include emphysema, congestive heart failure, pneumonia, pneumothorax, paralysis of the respiratory muscles, and even long-term use of barbiturates and alcohol. So the next respiratory acid-base disorder is respiratory alkalosis. This one is fairly uncommon. It is characterized by a high plasma pH and a low plasma carbon dioxide level because the respiratory system is eliminating too much carbon dioxide. So this would be called hypocapnia, or a lower than normal level of carbon dioxide in the blood. And the usual cause of respiratory alkalosis is hyperventilation, which would be an abnormally high respiratory rate. So this can be caused by physical stresses like pain or psychological stresses like extreme anxiety, like a panic attack, for example. Respiratory alkalosis usually corrects itself because low carbon dioxide levels reduce your urge to breathe until levels rise again. And if the pH gets high enough, a person will actually faint, so they'll lose consciousness. And while they are unconscious, they can't maintain the state of hyperventilation, and their breathing rate will adjust automatically while they are unconscious back to normal to recover the pH of the body back to normal. So other causes of respiratory alkalosis, and again, these are more rare causes, are exposure to high altitudes. So maybe you would see this in mountain climbers, especially the ones who climb the highest peaks like Mount Everest. So the very low oxygen pressure levels in the upper atmosphere promotes hyperventilation in an attempt to get enough oxygen, but at the same time it can cause you to lose too much carbon dioxide. Mechanical respirators, so ventilators in a hospital, if the breathing rate is set too high, that can cause this problem. And then brainstem injuries. So an injury to the brainstem means that a person can no longer automatically respond to carbon dioxide pressure changes in the blood, which is what normally adjusts your breathing rate. So here is the table from your textbook comparing and contrasting respiratory acidosis with respiratory alkalosis. So in respiratory acidosis, pH is decreased and carbon dioxide is increased. Respiratory alkalosis, pH is increased, carbon dioxide is decreased. And in both of these cases, bicarbonate ion concentration will normally be normal until you've had some compensation kicked in. And then in the case of respiratory acidosis, you would see increased bicarbonate levels as a compensation because your body is getting rid of hydrogen ions and holding on to bicarbonate ions. And you would see the opposite when you have compensation kick in for the respiratory alkalosis. So here is the picture from your textbook showing the homeostatic loop. So at the top, we're showing respiratory acidosis. So with respiratory acidosis, we are having hypoventilation, and that is going to increase our carbon dioxide levels. This is going to stimulate chemoreceptors. We're going to increase our breathing rate so that we can release some of the carbon dioxide, and at the same time, as a compensatory mechanism, the kidneys will be secreting hydrogen ions and generating bicarbonate ions. This is why you see an elevation in bicarbonate ions as a compensation for respiratory acidosis. And so the goal is to reduce the carbon dioxide, get rid of hydrogen ions in the blood, get rid of hydrogen ions in the urine and return the pH back to normal. The opposite would happen in respiratory alkalosis. You would have hyperventilation. This would decrease your carbon dioxide levels, causing an increase in blood pH. And in this case, we want to decrease our respiratory rate so that we hold on to more carbon dioxide as we make it in all of our cells. And then we can also have the kidneys kick in, and the kidneys will hold on to hydrogen ions and secrete bicarbonate ions, which is why you can see a lower bicarbonate ion level with compensation. And again, the goal of all of this is to increase pH back up to normal. So note that the ability to compensate by changing the respiratory rate depends on whether or not the primary problem involves the lungs. So for example, if your initial problem was because you're having a breathing problem, you may not be able to change the respiratory rate, but you can still get compensation by the kidneys. So now we'll look at metabolic acid-base disorders. So metabolic acid-base disorders occur when there is either an overproduction of acids, so you're adding extra hydrogen ions, or some condition affects the concentration of bicarbonate ions in the extracellular fluid, so either an increase or decrease in bicarbonate ions. So the first type of metabolic acid-base disorder is metabolic acidosis. This is the second most common disorder behind respiratory acidosis. Notice your two acidosis disorders are the most common because remember, we are always fighting against acidosis because our bodies naturally produce more acids. There are three major causes for metabolic acidosis. The first one is an overproduction of fixed or metabolic acids, and so this would be basically a production of too many hydrogen ions. Two examples. The first one is lactic acidosis. This can develop after either strenuous exercise or if your tissues have a prolonged lack of oxygen, which is called hypoxia. And when tissues don't have any oxygen, they can't do aerobic respiration in the mitochondria. And if they can, they switch to anaerobic metabolism to make ATP. But a byproduct of anaerobic metabolism is lactic acid. And lactic acid is an acid, so it adds hydrogen ions to the body fluids. So a lot of times with cases of the acute respiratory acidosis, you can also get secondary metabolic acidosis because when the person is not breathing, they also have hypoxia going on, which is also increasing levels of lactic acid in their bloodstream. Another type of example of an overproduction of acid is in ketoacidosis. This results from the generation of too many ketone bodies, and cells do this when they can't get adequate glucose. So this could occur during starvation when you're not able to eat enough glucose, or during uncontrolled diabetes mellitus where the glucose stays in the bloodstream and never is able to come into the cells, so the cells can't use it, so they have to switch to an alternate form of energy, and they end up making ketone bodies in the process. And so the ketone bodies are acidic, so they are actually ketone acids, and so that results in ketoacidosis. The second major cause of metabolic acidosis is if you have an impaired kidney ability to secrete hydrogen ions. So this could occur with kidney damage like glomerulonephritis, which was talked about in chapter 26. Diuretics can also cause metabolic acidosis because hydrogen secretion is usually linked to sodium reabsorption. Diuretics stop sodium reabsorption, and so they're doing that to help you lose water. So note that a diuretic is a medication that promotes fluid loss, but because they are stopping the sodium reabsorption, they inadvertently also stop the hydrogen secretion, so you end up with too many hydrogen ions left in the body. And the third major cause is severe bicarbonate ion loss. The most common cause of this is diarrhea, and during diarrhea, the intestinal contents pass through so quickly that there is no time to reabsorb all of the bicarbonate. So bicarbonate is secreted especially in the pancreatic buffers that come out in the duodenum, and the bicarbonate in these digestive secretions is normally reabsorbed before the feces are eliminated, but in the case of diarrhea, the contents are passing through so quickly you don't have time to reabsorb all of the bicarbonate ions, so you can end up with a severe loss of bicarbonate ions. So here is a flow chart for metabolic acidosis. So they're actually showing you two sides. So one side is when the problem is caused by an increase in hydrogen ion production or a decrease in hydrogen ion excretion. And the other side is shown if you have a decrease in bicarbonate ion concentration. But if you look closely, it doesn't matter whether the cause is an increase in hydrogen ions or a decrease in bicarbonate ions, the ultimate goal is very similar. So the ultimate goal is to increase the respiratory rate, which is going to remove carbon dioxide, which will remove hydrogen ions, and have the kidney secrete hydrogen ions into the urine and reabsorb or generate additional bicarbonate ions for the blood. So again, whether the problem is an increase in hydrogen or a decrease in bicarbonate, the ultimate goal of how to fix the problem is the same. The other metabolic acid-base disorder is metabolic alkalosis. This one is pretty rare. This occurs when bicarbonate ion levels become elevated, which results in a decreased level of hydrogen ion concentration. One possible cause of this is with repeated vomiting. So vomiting means losing hydrogen ions from the stomach. So hydrogen ions are part of the stomach acid. So if you vomit, you are losing this acid from inside your stomach. And to replace those hydrogen ions, more bicarbonate ions are released into the ECF through the chloride shift, as we covered in chapter 24. And so that will add bicarbonate ions to the ECF. Compensation includes hypoventilation, so slowing the breathing rate, and also loss of bicarbonate ions in the urine. So here's the flow chart from the textbook. So with metabolic alkalosis, you have a loss of hydrogen or a gain of bicarbonate ions. So your two goals are to decrease your respiratory rate, which will increase carbon dioxide, and you'll also increase your hydrogen ions by having the kidney reabsorb hydrogen ions and secreting bicarbonate ions into the urine. So here is the little table from the book that is comparing and contrasting these two. So with metabolic acidosis, your pH is decreased, your bicarbonate levels are decreased. With metabolic alkalosis, pH is increased, your bicarbonate levels are increased. And then in both cases, the CO2 is normally normal under the acute phase, but can change when you have compensation. So when compensation kicks in for metabolic acidosis, the CO2 levels can decrease because you have sped up your breathing rate. And with the compensation for metabolic alkalosis, your CO2 levels can increase because you have decreased your respiratory rate. So here is a nice little flow chart in your textbook and how acid-base disorders are diagnosed. So the first thing you would do is to check the pH of a person's blood. So the pH is going to tell you right away if you're looking at acidosis or alkalosis. The next thing you would do would be to look at the carbon dioxide levels. So you would see, are they either normal, are they decreased, or are they increased? And that would take you into the different branches on both sides of this chart. The next thing you would do would be to check the bicarbonate levels to see if they're normal, decreased, or increased. And then if you have narrowed it down to being a possible metabolic acidosis condition, remember with metabolic acidosis you can have either a loss of bicarbonate or a excess production of acid. So to differentiate between the two subtypes of metabolic acidosis, you can check what is called the anion gap. And the anion gap is a measure of the difference between negatively charged ions and positively charged ions in the blood. So if you want to get practice in figuring out whether a particular disorder is which type of acid-base disorder, we're going to do some of those in the activity, so in the in-class worksheet that accompanies this chapter. And so for our last section, we'll look at the effects of age. So we're going to focus on how your ability to maintain these balances changes as you age. So first, as you get older, your total body water content decreases, and this makes the fluid compartments more concentrated because you are losing water, so you have more solute compared to water. The glomerular filtration rate also decreases, so does the total number of functional nephrons that you have in the kidney. This can decrease the kidney's ability to regulate pH and fluid balance. Your ability to concentrate urine declines, so you end up losing more water in your urine, so you have to drink more in order to maintain fluid balance. You also secrete less ADH and aldosterone, which makes it even harder to hold on to fluids. So basically, as you get older, you need to drink more. You have a reduction in the vital capacity of the lungs. This reduces the ability for respiratory compensation, which puts you at an increased risk for respiratory acidosis. And arthritis can further contribute to this problem by limiting movements of the thoracic cage. So your ribs are not able to move to enlarge the thoracic cavity as much, so you can't take as deep a breath. Aging also affects many of the other body systems, which can have some effect on either fluid, electrolyte, or acid-base balance. And that is the end of Chapter 27. I encourage you to do the in-class worksheet and to watch the in-class activity video to get additional practice on acid-base disorders.